In the part 1, we have discussed about nucleic acid, which are the polymers of nucleotides. Further, we read that these nucleotides are phosphate esters of nucleosides composed of nitrogenous bases, pentose sugar, and phosphate group. Then we got to know about DNA, which is a double-stranded molecule having adenine, thymine, guanine, and cytosine bases. Then we saw its double helix structure given by Watson and Crick, and also its X-ray diffraction image by Rosalind Franklin. Then we have seen its different forms that includes A, B, and D. And finally, we have seen its major functions. Now in this part, we'll see the history of genetic material, that is DNA, and its experiment which proves DNA as a genetic material. Now let us see the history of DNA during 1800s. In 1866, Gregor John Mendel, who is known to be the father of genetics, discovered how traits are inherited in Pisum sativum and gave basic principles of genetics that includes law of dominance and law of segregation. And in 1900, Mendel theory were rediscovered by Hugo de Vries, Carl Correns, and Ehrenberg Teschemek. Now back then in 1869, Friedrich Mischer first discovered nucleic acids from the nuclei of pus cells that are leukocytes, which were from discarded surgical bandages, and called it nuclein. Now in early 1900s, Theodore Boveri and Walter Sutton gave the chromosomal theory of inheritance, where in 1903 Walter Sutton observed that diploid chromosome consists two morphologically similar chromosomes which are equally distributed during meiosis. That means transmission of genetic characteristics. Their findings are fundamental in our understanding of how chromosomes carry genetic material and how it passed from one generation to another. Now let's see the historical findings during 1900s. In 1944, Oswald Avery identifies DNA as a transforming material through the experiment on Streptococcus pneumoniae along with McCarty and MacLeod. Edwin Chargaff in 1949 discovered that the ratio of A to T and G to C are 1 or very close to 1. This ratio is called Chargaff's ratio or Chargaff's rule. The molecular basis of heredity was revealed when James Watson and Francis Crick elucidated the double helix structure of DNA in 1953. In 1977, Friedrich Sanger developed the rapid DNA sequencing technique, which was a chain termination method or dideoxy sequencing method. Now we see the different experiments that proves the DNA as a genetic material, which includes Friedrich Griffith experiment of 1928, Avery-MacLeod and McCarty experiment of 1944, and Alfred Hershey and Martha Chase experiment of 1952. Now the Griffiths experiment, which is also known as bacterial transformation or transforming principle. His observations were involved the bacterium Streptococcus pneumoniae. At that time, pneumonia was a serious cause of death in the wake of post-WWI Spanish influenza pandemic, and Griffith was studying the possibility of creating a vaccine. During the course of experiment, a living organism, that is bacteria, had changed into living form. That bacterium is found in two forms, that are smooth strain, whose cells produce capsule of polysaccharides, that is mucus, causing the colonies on agar to be smooth and rather shiny. This strain is virulent, pathogenic, and causes pneumonia. The second strain was rough strain, whose cells lack capsule and produce dull, rough R colonies. Both smooth and rough strains are found in several types and are known as S1, S2, S3, and R1, R2, R3 respectively. Now in this experiment, what Griffiths done, he injected the different strains of bacteria into mice. Then he found that the S strain killed the mice while the R strain did not. He further noted that if the heat killed S strain was injected into a mouse, it did not cause pneumonia, while when he combined heat killed S with live R strain and injected the mixture into a mouse and found that the mouse developed pneumonia and got died. Hence, Griffith concluded that the heat-killed S strain were responsible for converting live avirulent R cells into virulent S1 and called the phenomenon transformation. He called the genetic information which could be passed from dead S cells to R cells the transforming principle. Now the Avery-MacLeod-McCarty experiment. In 1944, Oswald Avery, Colin MacLeod, and Maclean McCarty revisited Griffith's experiment and concluded that the transforming material was pure DNA, not protein or RNA. These investigators found that DNA extracted from a virulent strain of the bacterium Streptococcus pneumoniae, also known as pneumococcus, genetically transformed an avirulent strain of this organism into virulent form. Their findings were included: DNA alone from S bacteria caused R bacteria to become transformed. Proteases and RNAs did not affect transformation, while the digestion with DNAs can inhibit transformation. Hence, they concluded DNA as a genetic material. Now, Avery-MacLeod and McCarty had shown that only DNA extracts from S cells cause transformation of R cells to S cells. To demonstrate that contaminating molecules in the DNA extract were not responsible for transformation, the DNA extract from S cells were treated with RNAs, DNAs, and proteases, and then mixed with R cell. Then he observed that the samples which includes no protein and no RNA shows transformation, while the sample which has no DNA shows no transformation. Hence, they concluded that transformation can't occur unless DNA is present. Therefore, DNA must be the hereditary material. Now, the Hershey-Chase experiment, which is also the transduction experiment. The Hershey-Chase experiment was conducted in 1952 by Alfred Hershey and Martha Chase that identified DNA to be the genetic material of phages. What is a phage? A phage is a virus that infects bacteria. It consists of a protein coat that encloses the double-stranded DNA. Hershey and Chase designed an experiment using radioactive isotopes of sulfur and phosphorus to keep separate track of the viral protein and nucleic acid during the infection process. They used the T2 bacteriophage and the bacterium Escherichia coli. The phages were labeled by having them infect bacteria growing in culture medium containing the radioactive isotopes sulfur 35 and phosphorus 32. Here Hershey and Chase then proceeded the experiment to identify the material injected into the cell by phages attached to the bacterial cell wall. When the phosphorus 32 labeled phages were mixed with unlabeled E. coli cells, they found that the phosphorus 32 label entered the bacterial cell, and the next generation of phages that burst from the infected cell carries a significant amount of phosphorus 32 label, while when sulfur 35 labeled phages were mixed with unlabeled E. coli, they found that the sulfur 35 label stayed outside the bacteria. Hence, Hershey and Chase thus demonstrated that the outer protein coat of a phage does not enter the bacterium it infects, whereas the phage's inner material consisting of DNA does enter the bacterial cell. Since the DNA is responsible for the production of new phages during infection process, they concluded the DNA as the genetic material of phages. Thank you.